Welcome, everyone. My name is Dr. Carmelo Thomas. I'm from the University of North Carolina at Wilmington, and I'm here today to talk about some experimental work that we've been doing that we expect will soon give us some exciting responses. For many years, I've been studying marine phytoplankton, in particular those species which are harmful or that produce bioactive compounds. They are organisms which naturally occur, grow in high densities, and often are expressed in the form of fish kills or intoxications of humans who've eaten contaminated shellfish, phenomena such as these. These are natural phenomena that oftentimes are beyond our particular control. Within our laboratory, however, we have been isolating and cultivating these organisms for many, many years. And along with me, Alexis Marti, my associate, has been working with us in recent years in doing some studies with a particular instrument that she's going to be describing later on in this presentation. One of the things that perhaps is of particular importance is to understand the fact that our environment has a very unique set of circumstances. The circumstances are these, and that is that the Earth's surface is covered, at least 71% of it is covered by water. And of that, over 90% is seawater or the ocean. The rest are smaller quantities that are located in ice caps and groundwater, and then a very small quantity of it, somewhere around 0.3%, is used for living organisms. The important point here is that this component of the ocean is one of the largest and most diverse ecosystems that we have, perhaps even more diverse than anything that's on the terrestrial component of the Earth. It's diverse in the sense that it has life of all types. It goes all the way from marine mammals and whales to all the way down to microscopic plants and animals called phytoplankton and zooplankton, and then even microbes. Of these organisms, it's the phytoplankton which are of particular importance. These are unicellular organisms that reproduce very rapidly, oftentimes referred to as plankton blooms, many of which are beneficial and required by all sorts of organisms for survival, but many of which are also harmful in the sense that they produce substances that usually causes these disruptions, these environmental disruptions, that we call harmful algal blooms. It's that particular group of organisms that we've been studying for a great deal of time, and it's these organisms that we focused in our culture collection in being able to grow in high concentrations to be able to study the compounds that are produced by them. Organisms from the sea have been studied by pharmaceutical companies for quite some time. These are usually things like sponges or soft corals that are oftentimes harvested in large quantities to be able to extract the bioactive materials that they produce. The problem with this is that, one, it's a finite resource. Some of these sponges occur only in certain places, and there's a limited number of them. And as a result, they are not a renewable source. On the other hand, marine phytoplankton grow very, very rapidly. They use small amounts of nutrients. They oftentimes can be grown easily in the laboratory. And by isolating single cells, we can produce millions and millions of copies in the form of dense cultures. As phytoplankton, however, they do have very strict requirements. They require light. They require nutrients of various kinds, plant nutrients, in very small amounts. And again, they require stable environments. They also require a condition where the pH and temperature doesn't vary a great deal from time to time. These organisms require a new type of technology to be able to grow. They produce these compounds in very minute quantities, and for the kinds of chemical elucidations for some of these compounds and studies, we require extremely high densities. In response to the special requirements that are needed for studying these organisms, we have developed a new technology in collaboration with ICAWorks, and basically is what we are calling the AlgaeMaster 10 and AlgaeMaster 100 for the growth under very strict controlled conditions in the laboratory of these very delicate organisms that we normally can isolate from the open ocean. Now my associate Alexis Marti will describe the units and the various features.
The Algae Master 10 and its sister, the Algae Master 100, meet the strictest of requirements necessary to grow microalgal cultures. This reaction vessel allows for an inert environment which can be sterilized through gas or steam, heat, or UV radiation. The vessel has a double-jacketed chamber which allows for water circulation by the connection of a circulating water bath. This water temperature can be controlled by an internal temperature probe in the unit, which is then connected to a control unit that can maintain the temperature of your culture to plus or minus 0.5 degrees Celsius. All internal parts of the unit are made from autoclavable material that contain no metal parts, which could potentially leak trace metals into your culture as a contaminant. The unit comes with a glass tube that contains a gas sparger upon the end, which allows for the aeration of the culture for the user to input gas, oxygen, or carbon dioxide if necessary. The mixing of the culture is done through this magnetic coupling, which is attached to the head of the unit. The shaft and the stirring paddles are created from Teflon material, which are also autoclave resistant. The stirring speed is controlled also by the control unit. The pH can be maintained through a pH probe. This is controlled by the unit, which can maintain the pH to 1/10 of a unit. The pH can be controlled through either gas diffusion by use of the sparger or through these two external peristaltic pumps. These pumps can be used to input acid or base buffers if necessary. Everything that you can do with this unit is controlled by this external computer control unit. The temperature, the lighting, which is done through these two LED panels, the pH, and the stirring.
The significance of this new technology allows us to be able to optimize conditions for growth in the laboratory, particularly for looking at the metabolic processes or products that are produced by these organisms. To produce the huge biomass which is required by the chemist to be able to examine some of these unique chemical compounds, and for testing for subsequent phases in the industry that's going to require growth of these organisms. The versatility of the Algae Master as a research tool is adaptable from a very small laboratory setting to a larger one in which you have multi-unit integrated systems and eventually a larger growth system. Thank you for your attention.